Ever looked at an amino acid chart and felt like you were just memorizing random letters? On the MCAT, it's not about memorization, it's about recognition and application. In this video, we'll turn amino acids into patterns you can actually understand, from structure and polarity to how they build functional proteins. By the end, you'll be able to quickly identify key properties and predict behavior exactly what the MCAT is testing. Let's make this one of your strongest topics. Amino acids share a common backbone, with variable side chains that determine their chemical properties and behavior. Every amino acid on the MCAT has the same core: an alpha carbon bonded to an amino group, a carboxyl group, a hydrogen, and an R group that makes each one unique. At physiological pH, remember the zwitterion form. The amino group is protonated as NH3+, and the carboxyl is deprotonated as COO, giving the molecule both positive and negative charges. The R group drives classification nonpolar, polar uncharged, acidic, or basic, and that classification predicts solubility, interactions, and placement within proteins. A helpful mnemonic is GAVLIM for nonpolar, STNQ for polar uncharged, and remember acidic are D and E, basic are K, R, H. Glycine is the exception with no chiral center, while all others, except glycine, are chiral and on the MCAT are assumed to be L-amino acids. Proline is another special case with a ring that restricts flexibility. On passages, you'll often infer how a mutation from one R group to another alters protein function, like swapping a hydrophobic residue for a charged one, disrupting folding. This concept ties directly into protein structure, enzyme activity, and even electrophoresis behavior, since charge varies with pH. Amino acid ionization states depend on pH and pKa, which determines net charge and isoelectric point, or pI. The key idea is that groups gain protons when pH is below their pKa and lose protons when pH is above their pKa. For a simple amino acid without an ionizable side chain, there are two pKa values, one for the carboxyl group and one for the amino group. As you titrate from low to high pH, the molecule goes from fully protonated to fully deprotonated, passing through a zwitterionic form. The isoelectric point is the pH at which the net charge is 0, and for simple amino acids, it's the average of the two pKa values. For amino acids with ionizable side chains like lysine, aspartate, or histidine, you must consider 3 pKa values and identify which two surround the neutral species. On the MCAT, expect titration curves where you identify buffering regions or equivalence points, or calculate pI quickly. A useful trick: acidic amino acids have low pI values. Basic ones have high pI values. This concept connects directly to techniques like isoelectric focusing and electrophoresis, where molecules separate based on charge differences that arise from pH relative to pI. Peptide bonds form through dehydration synthesis and exhibit partial double bond character that restricts rotation. When two amino acids link, the carboxyl group of one reacts with the amino group of another, releasing water and forming an amide bond. This bond is planar due to resonance between the carbonyl oxygen and the nitrogen, giving it partial double bond character. That means limited rotation around the peptide bond, which has big implications for protein folding. The backbone instead rotates around the phi and psi angles adjacent to the peptide bond. On the MCAT, you'll often be asked about how peptide bonds contribute to stability, or how enzymes hydrolyze them during digestion. Proteases like trypsin and chymotrypsin cleave specific peptide bonds, which is another favorite test topic. Also remember directionality. Peptides are written from N-terminus to C-terminus, and this matters when interpreting sequences or mutations. The rigidity of the peptide bond sets the stage for higher-order structures like alpha helices and beta sheets, which rely on predictable backbone geometry. Protein structure is organized into 4 levels, each stabilized by distinct interactions. Primary structure is simply the amino acid sequence, held together by peptide bonds. Secondary structure includes alpha helices and beta sheets, stabilized by hydrogen bonds between backbone atoms. Think C equals O to NH interactions. A classic mnemonic is that alpha helices are like coiled springs, while beta sheets are pleated. Tertiary structure refers to the full 3D folding of a single polypeptide and is stabilized by side chain interactions, hydrophobic interactions, hydrogen bonds, ionic interactions, and disulfide bridges between cysteines. Quaternary structure involves multiple polypeptide subunits coming together, like hemoglobin. On the MCAT, you'll often be asked how changes like pH, temperature, or mutations disrupt specific levels. Denaturation typically affects secondary and tertiary structure, but not primary. Also, expect connections to diseases like sickle cell anemia, where a single amino acid substitution alters protein behavior. Understanding which interactions are disrupted helps predict functional consequences. Protein folding and stability are driven largely by the hydrophobic effect and can be disrupted by environmental factors. In aqueous environments, non-polar side chains cluster inward away from water, while polar and charged residues face outward, creating a stable conformation. This hydrophobic collapse is a major driving force of folding, alongside hydrogen bonding and ionic interactions. Chaperone proteins can assist in proper folding, preventing aggregation. On the MCAT, denaturation is a key concept. Heat disrupts hydrogen bonds, pH alters, charge interactions, and chemicals like urea interfere with non-covalent forces. Importantly, denaturation typically does not break peptide bonds, so primary structure remains intact. Misfolding can lead to disease, such as prion diseases or Alzheimer's, which ties biochemistry to physiology. You may also see questions about how changes in environment shift protein conformation and activity, especially in enzymes where active site shape is critical. Always connect structure to function. If folding changes, function almost always follows.